Okay, good afternoon. You all know why we're here today. But one of the reasons why sleep was suggested by many people and certainly enthusiastically supported by myself is because sleep is one of those things that we almost take for granted unless we happen to have some disorder or affecting. But, you know, it's sort of like breathing and all kinds of normal activities. But it's a great mystery and an enormous challenge. And in this modern era, some windows are opening and doors are opening, and some clarity is coming toward helping to answer some of the age-old questions. I put on the website some material, but I also put a reference to this book. Now, normally we don't huckster books as part of the course, but Eric Kandel, the Nobel laureate for his work on memory, this is an amazing book which touches very much on consciousness. In fact, consciousness from the perspective of art in Vienna at the turn of the last century, music, the birth of cognitive psychology, the changes in medicine, and really some popular science level hardcore neurobiology. And so for liver doctors like me, this was a wonderful thing to read. So I put the reference on the website in case, you know, any of you are interested. It'll keep you busy for several months. It's taken me over 2, and I haven't finished it yet, but that's a reflection more of my limited upbringing rather than, but it's really written elegantly. So why, what, how, and when? And today's 2 speakers, we're fortunate once again in having here at NIH always an array of people in almost any major area of interest you can think of. And so today's speakers are going to be, first will be Carolyn Beebe Smith, who took her degree at the University of London and came here when you came to NIMH in 2000? In 2011. Okay. That's right. It says here you work with Lou Sokoloff and NIMH. Right. And she is a senior investigator in the Laboratory of Cerebral Metabolism. And as you will see, she is exploiting new technologies to study sleep and other disorders in experimental normal transgenic animals and their brains. And our second speaker is Susan Harbison, who, when I asked her why did she had an original degree in aeronautical engineer and then made a dramatic change and took a PhD and emerged with expertise in Drosophila, particularly taking advantage of the unique genetics of Drosophila to study problems related to sleep and other disorders. Susan joined the Heart Institute here in 2012 as an Earl Statman tenure-track investigator. For those of you who don't know, this is an extraordinary program that the NIH has, which is highly competitive, and it's really the creme de la creme that wind up coming here with a position as an investigator. So I want to just say a couple of words, because what went through my head when we were thinking of sleep was sort of a transition in time, which sets the stage for today's presentation. So time really starts, I think, in poetry and literature where death as a manifestation of sleep, eternal sleep, attracted the attention. Here, Prospero in The Tempest, our little life is rounded with a sleep. And if one reads at that time in history, the concept of death as a form of sleep and sleep as a form of death, those things were intimately linked. It's a very interesting question. They probably believed that when you went to sleep, your brain went dead. Very important concept. And then comes the area of the development of anesthesia, chloroform, ether. And people also went unconscious, although they maintained their vital signs if they didn't inhale too much. And what was that anesthesia? Was that the same as sleep? Is it the same as sleep? There's a huge literature that initially said it was sleep, induced sleep. And then there were others who came along and said, no, this is different. And then we get into now the late 19th century, where concepts of consciousness, sleep, and then later the introduction of electroencephalography as really the only tool aside from pathology. But there in pathology, the tissue's dead, so you really don't know what's going on. You may have known what happened. So it was really, I think, the electroencephalogram that showed the various wave patterns associated with people falling asleep, and the discovery of rapid eye movement sleep, and waking people up and discovering that's when they were dreaming. And this sort of set the basis of another important concept, and that is the brain is not dead when you're asleep. It's very much alive. But that may sound simple, but how it's alive is still the mystery today. And then beginning in the early 20th century and accelerating like crazy in the modern world is the development of cognitive psychology. And that's where this book, The Age of Insight, is a brilliant discussion of the role of the cognitive psychologists, including Freud and his colleagues, and artists and poets and writers and early neurobiologists in the recognition of emotions and consciousness and their relationship. And then, of course, we zoom up to the present time when cellular anatomy is now accessible in living tissues, more so and more so, through the use of anatomic, functional metabolic scans, a literal dissection of the central nervous system. And then that becomes coupled with, you move to another species, which we'll hear about, Drosophila in this case, where the genetics are all there. How can you use that to explain phenomena of sleep? This same theme is going to appear in a Walls lecture later on next month when Ulrike Eberlein comes and talks about alcohol studies in Drosophila and how they have enormously contributed to understanding things like alcohol tolerance, rejection, addiction, and so forth. So, enough. Beebe, are you speaking first? Okay.
On. Can you hear me? Yes? No? Okay. I'll project a little more. And let me know if it's not enough. My voice is a little rough. So I just want to thank Lynn for the invitation. This should be fun to get together and talk about sleep. And thank you all for coming. So I am going to, is this okay? I'm going to tell you a little bit about sleep in general, some of the just big ideas about sleep, and then eventually go into the study that we're in the middle. So we talked just now a little bit about, are you alive or dead when you sleep? Is that? I'm sorry. I hope that's not something okay. So when you're asleep, there's the characteristics are, there's the absence of motion, you have a characteristic posture, and a reduced response to sensory stimuli. It reduced, not no response. So, and it's immediately reversible. So that's the difference, right? There's the rub, right? Now, when we're people very interested in sleep are interested in a lot of aspects. One is there are many disorders of sleep. And so that, of course, is a big focus of a lot of sleep research. In addition, particularly in our culture, we're very interested in the effects of lack of sleep. And we all have personally experienced that to some extent. There are some extreme examples of what happens when you don't get enough sleep. And then there's kind of the academic question, which is why do we need sleep? What is it? What's happening to us when we're asleep? Why is it important? And this, of course, is a big, it's a grand, big research question. So some of the disorders of sleep. This. Okay. So the first two, insomnia and hypersomnia, is inability to go to sleep and stay asleep, or sleeping too much, are disorders of sleep, but they're also symptoms of something else wrong with your brain, usually psychiatric problems. So people who are depressed, for instance, can have either of these, can have either insomnia or hypersomnia. So some of the disorders of sleep actually can be symptoms of other disorders, and then we can ask, do they make that condition worse? You might imagine if you are depressed and then you can't sleep, you may become much more depressed. Another one that I'm sure you've heard about is restless leg syndrome. It occurs more in older people, where they have sensations of kind of itchy feelings in their legs and have to move, and then they wake up, right? So another one is bruxism, which is clenching your teeth when you're asleep. Sometimes when this is very extreme, it can wake you up, and of course it also can wear down your teeth, so your dentist will be talking to you about that if you have that problem. And the next one, sleep apnea, is another one that it's been a lot in the news. It occurs more in middle-aged men, particularly overweight middle-aged men. And what happens here is there is actually a collapse of the airways. There's loud snoring, which is one of the things you can detect with somebody who has sleep apnea, and they actually their blood oxygen level will go down, and that wakes them up. So these people can wake up 100 times a night. So you can see how that would interfere with your ability to sleep. And of course, narcolepsy is another one that I'm sure you've all heard of, where somebody in response to emotion then all of a sudden loses all muscle tone, collapses, and goes to sleep. So this is a particularly terrible affliction. An even worse affliction is one that, again, I'm sure you've all heard of. It's fatal familial insomnia. This is a disease that is an autosomal dominant inheritance where there's a mutation on the gene for the prion on chromosome 20. And this was first described in 1986 in an Italian family by Lugarisi and colleagues. And it's a progressive, untreatable insomnia. And people that are afflicted with this disease then develop this insomnia and will die within a fairly short time. And this can strike when somebody is in their 20s or when they might be fine until they're in their 60s. So what we see in their brains is that there's a characteristic degeneration, particularly in the thalamus, and also this spongiform encephalopathy, which you can see here in the thalamus and in the cortex. Here's the cortex and the thalamus, and that's accompanied by gliosis. So this is a particularly dreadful disease that fortunately is very rare. Now we all know from personal experience again that there are many effects of sleep deprivation. There are some interesting studies in rats, for instance. Rechtschaffen has shown that in rats that are totally deprived of sleep, they will die in about 3 weeks. So it's, again, fatal, not sleeping. And there was a story from that I remember from my youth, which maybe some people in the audience may remember, of a DJ in New York City named Peter Tripp, who he was very popular with everybody. He was the guy who invented the top 40, and he in order to raise money for March of Dimes, said he would stay awake for over 200 hours. And he actually had a little glass booth in Times Square, and it was a big production. There was a lot of publicity about it. And he started to show signs of psychotic behavior after a few days of that. He did do it. He stayed awake for 200 days. He had to have some medication to keep him awake. I presume, I don't know what they gave him, but anyway. The interesting thing afterwards is that he was never the same.
He became president, is that
right? Right, he became president, yes. He lost his job, he got involved in crime, his marriage broke up. I mean, his life kind of fell apart. I don't know whether that had anything to do with the sleep deprivation, but just to say that was a dramatic example in the news of somebody who had starved himself of sleep. Now, for most of us, we go around, I think, with our sleep tank on about a quarter, or maybe a half, some of the good ones of us, but we're usually not very good in our culture about sleep hygiene. We kind of undervalue sleep. And there are some pretty important consequences of that. There are psychological ones, which I think you probably know about. You reduce your performance, you have lower reaction time. You also increase your risk for depression and anxiety, and you also suppress your immune system. And then you increase your risk for a number of diseases and even for obesity. So clearly sleep has an important function. If just being deprived of sleep is going to produce such effects. What sleep, as Wynn said in his introduction, is not kind of a single state. There are numerous stages of sleep, and I'm not going to make this into a lesson in polysomnography so you can identify all the stages, but this is just to illustrate. These stages can be defined by polysomnography in which there are recordings of the electroencephalograms. So there are electrodes put around your scalp, and your electrooculograms, so that now they're under your eyes to monitor your eye movements, and electromyogram to look at your muscle tension, and there it's usually under the chin. So when you're awake, you have these characteristic high-frequency waves in the EEG, lots of eye movements and lots of muscle tension. So when you first fall asleep, you go into stage 1, which is kind of a transitional stage, and the EEG becomes a little lower frequency. Eye movements are slow, and the muscle tension is medium. In stage 2, you're getting a little deeper. Now the EEG has some interesting waveforms, these K complexes and sleep spindles, which just indicate that you're getting into a deeper sleep. Eye movements have ceased, and again, the EMG is kind of medium tension. When you go into stages 3 and 4, that's now you're into a deep sleep, a slow wave sleep. And here, the EEG has these characteristic delta waves that are of low frequency and high amplitude, and there are more of them in stage 4 than in stage 3, but they're very similar in the two. You see more of these. And again, eye movements have ceased, and the EMG is low. And finally, REM, which was the last stage to be discovered. And there, the EEG looks a little more like light sleep or awake, and you see these rapid eye movements, and one of the most characteristic things is the muscle tension is just about gone, so they're very, very relaxed. So just to kind of summarize what happens with sleep over these stages. So when you're awake, you have this desynchronized firing of neurons. When you go into non-REM sleep, the firing of the neurons is more synchronized. So you've gone from a state where neurons are all kind of doing their own thing in your conscious brain to a state where they're much more firing together and a little more oblivious to what's going on outside. And in REM sleep, they're desynchronized again. So this is a state that is quite different from the non-REM sleep. And over a night, you go in steps. This is hours of sleep. So you go in steps to this deep stage 4 sleep, and then have a few climbs back up to REM. So in the beginning of the night, you spend a bigger proportion of time in deep sleep, in slow wave sleep, stage 3 and 4, and some smaller time in REM. And then as the night progresses, you start to spend more time in stage 2 and REM sleep. So this is kind of the progression of these waves of sleep over the nighttime. Now, over your lifetime, sleep also changes. So we all know that babies spend a lot of time asleep, maybe 16 hours a day asleep, and about half of that is in REM and half in non-REM. Prenatally, they spend even more time in REM. And then as they develop during early childhood, the REM decreases, but actually the non-REM, the component goes up. And this, in the first few years of life, stays pretty constant, whereas REM is decreasing. As we get older, we spend less and less time asleep and less time in REM. So there are several processes that drive sleep. One is circadian, so this is just the circadian 24-hour clock. And the other is a homeostatic drive, like any appetite. It's just like being hungry. When you're awake for a long time, you get sleepier and sleepier the longer you are awake. So this just illustrates that over the awake and asleep periods, that you have this circadian pattern, but the homeostatic drive, the longer you're awake, the bigger your drive to sleep, and then as you're asleep, that diminishes and you wake up. So the circadian drive is through a part of the anterior hypothalamus. There are these two little nuclei in the anterior hypothalamus called suprachiasmatic nuclei. And they are connected to the retina. There's a synapse from the retina to the SCN. So light will stimulate the retina and then stimulate the SCN. And that will stimulate some neurons in the lateral hypothalamus, these hypocretin or orexin neurons, that then act to activate brainstem activating systems. And this just shows the circadian pattern of an animal that is showing his running rhythm, and he's entrained to the light, so he in fact is quiet in the light and running in the dark. The homeostatic drive, one of the major players is adenosine, and this is a product of cellular energy metabolism, and it accumulates during wakefulness, and then it also acts as a neurotransmitter. So you can see how adenosine could be something that would act on that drive to sleep as adenosine is going up. And this is just to show you that there are these activating systems in the brain that are sort of feeding up from the brainstem or the pons to activate the cortex, the thalamus, and sort of keep you alert, aroused. And there are three major systems: the reticular activating system, which is a cholinergic system, from the locus ceruleus, which is a noradrenergic projection, and the dorsal raphe, which is a serotonergic projection. Oops. So we said at the outset that one of the big questions is why do we need to sleep? It's clear we need to sleep, but why? So one of the ideas is that during wakefulness, there's an accumulation of metabolic products that have to be cleaned up, taken care of, and there's also maintenance that needs to be done on the nervous system just to take care of wear and tear, just as you would a road system. And maybe this is best done when the brain isn't doing other things. So that's called the restoration hypothesis. It's a very simple idea, very difficult to prove. So, and another idea that has been discussed a lot in the last 10, 15 years is the influence of sleep on synaptic remodeling. And so this pertains mainly, again, to the nervous system, in that in the nervous system, one of the key attributes of the nervous system is this plasticity. So plasticity is the ability of the nervous system to adapt to changes in environmental input. And in development, plasticity is, of course, very important for getting pathways organized. But it doesn't only occur in development, it also occurs in all of us, or we wouldn't learn and remember anything. So plasticity is a very important process. And what we're learning is that there is an influence of sleep on plasticity. And so I'm going to focus all the rest of my remarks on this issue. Now, to talk about developmental plasticity, one of the key systems that's studied, because it's a very robust system, is plasticity of the binocular visual system. So animals with binocular vision, like a cat, a monkey, us, we have input from both eyes, and that input, if it originates, say, in the left part of the visual field, will go through both eyes. The input goes through the thalamus and then to the primary visual cortex. But the input from the two eyes remains segregated all along this pathway, so through the thalamus and in the cortex in the form of these ocular dominance columns. So from the left visual field, it will activate the right primary visual cortex, and the left visual field will the right visual field, will activate the left. Well, what we've learned about this system, and this is largely from the work of Hubel and Wiesel, for which they won the Nobel Prize, is that during development, if you block the input from one eye during the time that this is developing, you will actually end up with a cortex that is responsive now only to the open eye. And people have shown this in cats and in monkeys. So we in neuroscience use this system a lot to study plasticity. And Marcus Frank's lab looked at this process in cats and in kittens, and he asked the question, does sleep have any effect on this plastic response? So I'll just tell you just for a second what these are so that it doesn't become overwhelming. But so these are essentially their percent recording of neurons that are responding to light flash from going through one eye or the other, and he's indicated here. This is the open eye, this is the deprived eye. So he took a group of cats, and they have binocular vision to start with, and then they had 6 hours of monocular vision while they were awake in the light, so they're getting visual stimulation. And then he looked at what this did to this histogram of ocular dominance. And you can see there's a little tendency for dominance to be higher in the eye that has visual input and lower in the deprived eye. Then he took another group, did the same thing, but then at the end of the 6 hours of wakefulness in the light, he put them in the dark and let them go to sleep. And then after 6 hours did a recording session. And now he's shown that there's this shift that's pretty dramatic, that these cells now more cells are responding to the open eye and fewer to the closed eye. So the next question is, well, is that just because they had another 6 hours? And so he took another group and studied them while they remained awake. So he kept them awake, but in the dark. And here you can see he didn't get this effect. So it isn't just time. It seems to have something to do with sleep. And he took a fourth group and kept them awake, but in the light. So now they've had 12 hours of monocular stimulation. And here he sees a pattern that's rather similar to what he saw with the sleep. So that suggests that the sleep enhanced the effects of that preceding period of monocular deprivation, and enhanced that plasticity response in the visual cortex. So the next question that his lab asked was, Well, what processes does that depend on? And one of the first things that most of us who are interested in plasticity think of is protein synthesis, because we know that plasticity is usually blocked by protein synthesis inhibitors. So he studied the same system, but he used an inhibitor called rapamycin, which blocks the mTOR pathway, which is a regulator of protein synthesis through a signaling pathway. And so in this series of histograms, first you can see what it looks like when a kitten is studied that has binocular vision. So you see what the histogram looks like with this normal distribution. And then he had a group of animals that got vehicle, and another group here, or two groups that got rapamycin that was applied to the cortex. And when he did monocular deprivation only, whether they got rapamycin or not, he really didn't get much of a change in the histogram. But when they slept, during the time that they got the vehicle or rapamycin, you can see that in the vehicle-treated animals, he got that same shift that he saw in the previous experiment, but when he did the same thing but treated them with rapamycin, it was blocked. So the conclusion is that this process that's going on during this plasticity has something to do with, is involving protein synthesis. So that was all taking place in the developing nervous system. But as we said, this happens in all of us, whether you have a mature nervous system or not. And so there are a lot of people who have been studying the effect of sleep on memory consolidation in the mature nervous system. And a lot of this comes from the work of Robert Stickgold and Walker and colleagues. And they use, and this is a very simple slide, but they use this test called the texture discrimination task, which I'll explain a little bit more as we go on. But the point here is that with this task, if they train them on the task, let them sleep, and then test them afterwards, that they did better the longer they slept. So this is something that all students should be shown. That don't stay up all night and study for the test. Go study all along and then sleep the night before. Right? So we're very interested in a combination of these issues. So we're interested in plasticity, and particularly in the adult brain. And we're interested in how protein synthesis is involved in that process during sleep. So I'm going to tell you about a study that is ongoing. I don't have final results, but I'll tell you some preliminary results. And this is a study that I'm doing with Tom Balkin at the Walter Reed Army Institute of Research and Dante Piccioni at NINDS, who are both sleep experts, Kathleen Schmidt, who's a kinetic modeler, and Ina Luteev and Adriana Pavletic, who are our physicians who are working on this. And many others are helping with it, but these are kind of the primary people. So we're asking the question, is the sleep-dependent improvement on the performance on that texture discrimination task, is that accompanied by changes in protein synthesis in the parts of the brain that are undergoing this plastic change? So we're studying healthy volunteers. They're young people. They're people with no neurological or psychiatric diseases, and, well, I say no sleep issues. I mean, people who don't have problems sleeping, might not have problems sleeping while we're doing a PET scan. So I haven't mentioned PET yet, but that's how we measure protein synthesis, and that's how we do this study. We've devised a way of measuring protein synthesis with positron emission tomography in human subjects, and that involves injecting a radiolabeled tracer amino acid, and then through kinetic modeling and the dynamic scan process, we actually can come up with a rate of incorporation of the amino acid into protein in all parts of the brain. So our procedure, because they have to sleep during our PET scan, we have to sleep deprive them before they do the study. And then we train them on the texture discrimination task in the morning. And then they get a PET scan with or without sleep. So we have one cohort that sleeps, one cohort that we continue to keep awake. And then they're retested on the texture discrimination task. So just to show you a little more about that task, so it's a computer task, so they're watching a screen. The first screen has just crosshairs in the center, and they're told to focus on the center of the visual field and watch that crosshair. Then they get a blank screen, and then they get the test screen. So these are some examples of test screens. And the test screen has a letter, L or a T, in the center of the visual field, and they're told that they have to identify what that letter is. And then we have these lines, and here these 3 lines are oriented vertically, and here they're oriented horizontally. Here, this side, they're in the left, and here they're on the right. And we tell them that what we want them to do is tell us what's the letter in the center of the field, and are these lines oriented vertically or horizontally. So they see that screen for a very short time, and then they get this screen, which is just a math screen, and then a blank screen, and then they answer. They put in their answers. And what varies is the interval between these two. And in the beginning, they get a longer interval, and it gets progressively shorter. And we look at that to decide their performance. So just, again, to explain the experiment, to go back to this diagram, so we can separate the 2 hemispheres. We can measure protein synthesis in both hemispheres. And if we always put the stimulus in the left visual field, we will be addressing the right hemisphere, and if we put it in the right visual field, it's here, we'll be addressing the left hemisphere. So we have an internal control. So this just shows you the PET scan. So we do our scans on either awake or sleeping subjects. They're monitored for polysomnography during that scan. And this is the high-resolution research tomograph, which has a spatial resolution of about 2.5 millimeters. So to start the scan, once the sleeping subjects have gone to sleep, we inject the radiolabeled tracer, which is 1C11 leucine, and we follow its disappearance in the arterial blood over the whole course of the scan. At the same time, we're scanning the brain, and you can see the tracer in the beginning is very low, then it's taken up into the brain, and then it's cleared. And what's left is mostly what's in protein. And we do an MRI to get the anatomy, and then we can identify all the regions of the brain and transfer those outlines to the PET image and get the rate. So first we wanted to know, well, if we sleep deprive these guys, can they do the task, and can they show improvement after a nap? So we just compared a sleep deprived group with a control group, a sleep sated group, and they all had a nap between the morning administration of the test and the evening. And I should be sure to point out that with improvement, this score goes down. So they all showed improvement. So that was reassuring. And now in our cohort of subjects that we've studied so far, we can see that those that were not permitted to sleep during the PET scan, were not permitted to nap, actually did worse on the second administration of the test. And those that did get a nap did slightly better. And as I said, we don't have final results, but I just wanted to show you a picture of what results in some of the subjects look like. So this is the primary visual cortex. The cuneate gyrus, it's here, left and right, and the lingual is below. So the lingual is responding to the upper part of the visual field, the cuneate the lower, and we always address the lower part of the visual field. In this subject, he was stimulated on the right, in the right visual field, so the activation, and it's not an activation like we usually think of it, this is an increase in protein synthesis, is occurring in the left hemisphere. So this is the result we hope that will be consistent throughout. We don't know yet, but that's what we're looking for sure. And that would be, I mean, if that holds up, that would be a direct demonstration of an increase in protein synthesis during memory consolidation task. So just to sum up, I think I've talked a little too long. I'm sorry, Susan. I'll let you take over. I just kind of go over. We talked about definition, disorders, and sleep drives, and then we got into plasticity and memory, and now our study, which is to determine if there is a protein synthesis dependent aspect to this sleep dependent memory. So thank you very much.
You mentioned the different stages of sleep. Have they done correlations of cerebral fluid chemicals to those different stages to help sharpen those definitions? Or is it just primarily some of...
I mean, like correlations with it, like adenosine levels, and is that what you mean?
It changes in this, yes.
I don't know. Do you know, Dante, if they have?
If I may add a little something to the question, if I may. So yes, adenosine levels have been measured with microdialysis in non-human animals, and adenosine levels actually increase. Increase during wakefulness. So that is the correlate of the need for sleep, and then those adenosine levels then decrease during sleep. And this says that adenosine acts as a sleep regulatory substance because it is behind the drive for sleep. But maybe your question also might have been getting towards neurotransmitters.
Yeah, neurotransmitters, and I mean, we classify RAM, non-RAM.
Yeah. Yes.
Go ahead.
Would you like to?
Go ahead.
Would you like to address the question?
Sure. The question was what neurotransmitter changes happen in the brain during a night's worth of sleep, and that's really under a lot of study right now. We do know that, so in fruit flies, which I'll tell you about in a minute, that dopaminergic levels, when they go up, increase activity in the flies, and when they go down, increase rest. But it's a study, it's a rather hot topic in sleep because think about the experimental challenges involved. Carolyn's got a really wonderful system to measure these levels of protein in the brain, but how would you measure neurotransmitters during sleep? Even if you're using an animal, you're talking about you have to sacrifice the animal. Well, aren't you changing things while you're doing that? You know, I've heard of people microwaving them, boiling them, and on the other end, freezing them in nitrogen, so it's not easy to do.
So let me ask you a general question about PET scanning and MRI. So you show evidence of enhanced protein synthesis in a certain area of the brain associated with a visual phenomenon. Now, the visual phenomena is an incredibly complicated business involving not just the optic system and the geniculate body and then feeding into the cortex and all the other ramifications. So it's a whole network. It's not a camera. How does that play into the fact that you would localize this to a very specific area in terms of that's where the primary effect is? Maybe it's the secondary compared to the whole cascade of everything that's happening, or am I naive about it?
Mystified by. So, no, it's a very good point. And of course, in our study, we're focused on the primary visual cortex because this is that's the place where there is the consolidation of the information from that very simple task. And this is, remember, not a complicated visual task. It's a pretty simple thing, locating whether these lines are like this or like this, right? I mean, so we're focused on that area, and that area, it's been shown in MRI scans, is an area that is activated by this task and even is reactivated during sleep after training on the task. So it's that's another very interesting observation. So we think we're looking at a particularly pertinent area, but we're not looking at everything in the visual system. So.
Biochemical or pharmacological differences during, say, REM sleep that I guess you can only speculate at this moment. There used to be a drug on the market called Quaalude, which was there used to be a drug called Quaalude, which worked so well that they took it off the market because people were enjoying themselves immensely. And that was supposed to produce only REM sleep. Is that possible or not? That was what was claimed. I don't know.
What is a Quaalude chemical? Oh, God. Yeah, I don't know. Does anybody know what a Quaalude is?
Susan, do you have a comment about Quaaludes? Huh? Yes.
So I think this goes back to the question of a little bit about neurotransmitters. And first, I think it's important to mention that in general, this is a very general statement. I have to emphasize that, in echoing the earlier comment, the earlier response about neurotransmitters, and that's a difficult area. But generally, during non-REM sleep, you get progressive decreases in serotonin, norepinephrine, and then during REM sleep, you have the nadir of these levels of neurotransmitters, or the firing rate, at least. If, both kind of studies have been measured in non-REM sleep. And then in REM sleep, what's unique is that they stay low, but you have a increase in acetylcholine. So that kind of makes it a unique state. So you might say, well, to get to your question, you'd say, well, what drugs simulate acetylcholine release? And sure enough, those drugs do increase REM sleep, but that's just one example. So if you stimulate acetylcholine, you get increase in REM sleep, and if you inhibit it, then you get decreases, generally.
Okay, well, we'll have time for more questions after Dr. Harbison is. Oh, Susan?
Okay, can everybody hear me okay? Okay. I'd also like to thank Wynn for the opportunity to speak with you today. We're going to do something a little bit different. We're going to talk about genetics in Drosophila. So as Carolyn very nicely pointed out, there are a lot of health impacts of curbing your sleep or depriving yourself of sleep fully. And sleep disorders also lead to problems with your health. So, for example, she mentioned that curbing your sleep can lead to a higher incidence of obesity, diabetes, and cardiovascular disease. And sleep disorders like insomnia are a hallmark of depression, as well as sleep apnea being a risk factor for hypertension. And so we want to know how sleep impacts human health, but we really don't understand the function of sleep. And there's some challenges in human sleep studies. One is that simultaneous measurements are only possible in identical genotypes if you have monozygotic twins. So this reduces your statistical power in a study because you can't, for example, control the environment that people experience. We all experience different environments, and you probably are very well aware that sleep, your environmental conditions can affect your sleep. You sleep a lot differently the night before your thesis defense than when you're on vacation at the beach, for example, right? So those are two sort of extremes. But you can understand that when you're doing a human study and you're sampling members of the population, you could be getting them at one of their good times or one of their bad times, and there's no real way to know that. And then finally, once you come up with candidate genes for human sleep, they're very difficult to test. It's a real challenge to test them in humans simply because the mutations that you might be looking for may not be present in the general population or not at a high frequency to where you can definitively claim that a certain gene affects sleep or not. So that's where Drosophila have some real advantages. That's because we can take outbred populations or groups of flies and inbreed them completely, and this inbreeding allows us to create genotypes that are basically identical within a line of flies. So every fly that you test in that line has the same basic genotype, and you can make those measurements simultaneously. So that's a real advantage. Another thing is that we can completely control the environment. I've got temperature and light-dark cycle, but we control also the food, the mating status, their crowding, how many other flies they have in the vial with them, and all of these things can impact sleep. And we have some genetic tools in Drosophila. There are tools like the Gal4 UAS system, which allows us to drive gene expression in both a spatial and temporal fashion. And we also have mutations and transgenics as well as deletions that can be tested. So if you have a region or a gene of interest, it's pretty easy to just order flies from the Drosophila Stock Center and then test your mutants. So Carolyn went over the characteristics of mammalian sleep, and I'll just touch on them briefly here. She mentioned an increased arousal threshold. That's a reduced response to external stimuli. Homeostatic regulation, which means that when you deprive yourself of sleep, obviously you're tired the next day, so you want to make up the sleep that you lost. Sleep is spontaneous and happily, as she mentioned, reversible. And it tends to be a relatively stable characteristic within a species. So in the year 2000, two different groups showed that flies have all of these same behavioral characteristics of sleep. And so, including sleeping in the bed. And so flies can be used as a powerful model organism for the study of mammalian sleep. So we measure sleep in flies using this Drosophila activity monitor. There are glass tubes that fit into these little holes, and there's a single fly in each glass tube. So for each monitor, we measure 32 of these. And the monitor has an infrared beam that bisects this tube. So when the fly walks back and forth, he breaks the infrared beam, and that registers an activity count, and all of that is recorded on a computer. Then you get a data file, which gives you the number of activity counts per minute of recorded time. And I don't have time to really go into why this is, but if you can ask me at the question. 5 minutes without an activity count is defined as sleep in flies, and that's different from just a transient rest period of 1 minute. So if we have all of these measures for the flies, we can calculate the sleep duration, the number of sleep bouts or naps that the fly takes, the average sleep bout length during the day or night, and the waking activity, which is the number of activity counts or beam crossings per minute of recorded time. And so to approach the study of sleep using systems genetics, we start, first of all, with a wild-derived mapping population, and I'll tell you about our population in a minute. We measure the sleep phenotypes on these flies, and then we obtain DNA sequence for all of the individual lines in the population. So we may have single polymorphisms. You may hear me refer to them as SNPs. So that's a single polymorphic difference. We also measure insertions and deletions and characterize those as well that may be present. And we measure the transcriptional level using RNA-seq. So then we can associate our DNA sequence polymorphisms with our phenotypes in order to identify quantitative trait loci, or QTL. We can also regress the transcript abundance on our phenotypes in order to identify quantitative trait transcripts, or QTT. And then we can also associate these polymorphisms with the transcript abundance to identify expression quantitative trait loci. So these are the polymorphisms that influence the transcript abundance. So I'm going to talk today about the first arm here, which is associating DNA sequence polymorphisms with sleep phenotypes. And so to do this, we use the Drosophila Genetic Reference Panel, or DGRP. This is a wild-caught flies from Raleigh, North Carolina, that were inbred by a full SIB mating for 20 generations. So each line of flies, which I have here as a solid color, has an identical genotype. But between the lines, there's a lot of genetic variation, and that sets up an ideal scenario for mapping our phenotype. Just for the sake of time, I'm going to focus on night sleep and day sleep. So that's the duration. So when I just say night sleep or day sleep, I'm talking about the duration in minutes of time that the fly spends asleep. So if we look at a histogram of line means from the Drosophila Genetic Reference Panel, with the male line means in blue and the female line means in pink, you can see that there's an enormous amount of variation. So if you think about the night period in flies, it's 12 hours, so that's 0 to 720 minutes. We have a line that's around 90 minutes, females, and these males here are over 11 hours. So it's quite extreme. And broad sense heritability here is quite high, indicating that there's a large genetic component for this particular trait. Day sleep is also highly variable. You notice the difference in the pattern of the histograms here. The females sleep a lot less during the day than the males. The males sleep a lot more. In some cases like this, these guys sleep pretty much all day. We also noticed something that was really interesting in our lines of flies when we plotted every fly in the experiment. So the females here are shown in pink and the males are in blue, and this is every fly in the experiment. The horizontal lines here are each a line of flies from the Drosophila Genetic Reference Panel. So there's 168 of these. And what you can see here is that if we order this from longest sleeping to shortest sleeping for the night sleep and the day sleep, we see that the long sleepers are far less variable than the short sleepers, which is pretty remarkable. And that says a lot about how much variation you can get in flies of an identical genotype. So you can see these guys really are quite extreme in their variation, but their mean sleep is quite low. And we can calculate this particular level of variation as the coefficient of environmental variation, or CVE. So that's just the standard deviation of a line divided by its mean times 100. So if you have a long sleeper line here, which is fairly consolidated, I guess you would say, and it's a phenotype, then you can see that the CVE is relatively low, and we say that those flies are robust to random environmental perturbations. If you look here at the short sleepers, they have a broad range of sleep phenotypes. Their CVE is much higher, and they're plastic, so they're relatively sensitive to environmental changes. So it turns out that this coefficient of environmental variation is also quite variable in the DGRP. You can see that the males here in this particular group are relatively insensitive, and so are these females. And then we have some very sensitive females out here. So they're sensitive to changes in the environment. And the same is true with day sleep, with the males here being a little bit less sensitive than females. And that might explain why they're able to sleep so much during the day. They're relatively insensitive to different environmental changes, like the light stimulus, for example. It's a big one. It turns out these measures are correlated if you correlate night sleep with its respective coefficient of environmental variation. The correlations here are pretty high. And so what that tells us is that if you find a polymorphism that affects mean night sleep, it's likely to impact the variation in night sleep as well, and the same for day sleep. So we did a genome-wide association on these lines of flies. I'm just showing you a kind of a cartoon here, just to remind you that each of the lines are homozygous. So for example, at a particular SNP, we expect to see an AA or a GG. There's no AG, there's no heterozygote. So that's another thing that makes the analysis of these flies less complicated than it does in the human situation where you'd see a lot of heterozygotes. If there's no pattern with a particular SNP and our sleep phenotype, there's no association. If there's a clear pattern where long sleepers of a particular genotype have a particular genotype and short sleepers also have a particular genotype, then that would be a significant association. So there are a lot of polymorphisms in the Drosophila genetic reference panel. It's been fully sequenced. And here I'm just showing you the distribution. These colors here represent the 5 chromosome arms of Drosophila. And then this is the count in millions of sites for intergenic and intronic polymorphisms. And those polymorphisms that are synonymous, missense, nonsense are in the 5 or 3' UTRs. And what that means is that these guys are really within the gene region and these guys are outside of it. And so you can see that the polymorphisms outside the genes are far more numerous than the ones inside the genes. And of course we expect that due to natural selection tending to select against polymorphisms inside the coding regions of genes. So there's 2,490,165 polymorphisms and that we associated our sleep phenotypes with. And we found that many SNPs are significantly associated with night and day sleep. Here I'm going to emphasize night sleep because we did the verification on those, and so I'll emphasize that more in a minute. So we found 160 SNPs in 53 genes for night sleep. Low-frequency SNPs, by this I mean about 5% minor allele frequency, so most of the population had the major allele. So most of our SNPs are these kind of low-frequency SNPs. And there's 18 human homologs for night sleep and 43 for night sleep CVE. For day sleep, at our threshold level, we didn't find any SNPs, which is a little depressing. And for day sleep CVE, we found 71 SNPs in 23 candidate genes. Again, these were predominantly low-frequency SNPs, and there were 4 human homologs. So most of our SNPs overlap between night sleep and night sleep CVE. In fact, all but one gene pretty much overlap between these 2 traits. So that again reflects that high genetic correlation between the 2 traits that I mentioned before. And so that's good. That's what we expected to find. And a lot of these, several of these SNPs rather, are found in homologs of human sleep genes. So there was a genome-wide association study done by Dan Gottlieb's group in the Framingham population. And he looked at daytime sleepiness, usual bedtime, and sleep duration. And this was done with a diary assay where they you record basically your sleep patterns over a period of about 7 days. And so we have 4 genes that overlap with the genes that they found in that study. And there's also been studies of narcolepsy, restless leg syndrome, and sleep apnea where we found some of our genes overlap. And so this is quite promising because it suggests that the genetic basis of sleep is conserved between flies and humans. So I just showed you a lot of SNPs that were associated with sleep. And in human studies, this is where the real difficulty begins. You might find some SNPs that you think are interesting, but how do you go about proving them? Well, with flies, we can take an artificial selection approach to confirm that these SNPs from our genome-wide association study actually affects sleep. So to do this, we have to start with a highly variable population of flies, and I'll show you how we created this. And then we can select for short sleep, for long sleep, and maintain an unselected control. And so we're going to measure sleep in each generation to do that selection. But of course, we want to know what's happening to the underlying genotypes. And then once we get the genotypic information, we want to associate the sleep with the genotypes each generation. And that will allow us to verify our SNP candidates, hopefully. So that's the hope. So we began with the 10 most extreme DGRP lines. On the left half of the graph are our short sleepers. And here we're going to do the selection on night sleep. So these are the kind of reddish purple bars that shows you night sleep. But I've also got day sleep in this lavender color and 24-hour sleep in this sort of bluish purple color. And that's just to show you the contrast, the difference. So even if you look at 24-hour sleep, the short sleepers sleep less than the right side of this graph, which are the long sleepers. And these guys here are almost uniformly sleeping about 11 hours at night on average. So they really are long sleepers. So we take those lines. Here I'm just showing you kind of a chromosome schematic of the flies. And the long sleeper SNPs are in the black color, and the short sleeper SNPs are in the black color, and the long sleepers are in the yellow. We can cross these together in a full diallele. So we take the males of every line and cross them with the females of every other line. And we can take the progeny of that and then allow random mating to occur. And we did this for 21 generations in flies. So that's each generation is about 2 weeks, so it's close to a year that we just allowed these guys to mix. So this gives us a highly variable mixed population. So you kind of see what we're doing now. We've taken the extreme lines and we're jumbling up all those genetic backgrounds now, and then we're going to select for long and short sleep and see what we get out at the end. So it's a nice way to replicate populations. So we split this mixed population into the replicate selection populations. We had 2 long sleeper populations, 2 short sleeper populations, and 2 controls. And then we measure night sleep in 100 males and 100 females from each population. And then we use the longest sleeping 25% as parents for the next generation for the long sleepers, the shortest sleeping 25% as parents for the next generation for the short sleepers. And for the control, we just chose 25% at random. So we're looking here at sleep characteristics in our replicate populations prior to selection. And the only thing I want to show you here is that for our control, our long sleeper and our short sleeper, that there wasn't any difference in night or day sleep that was statistically significant. And that's good. It means we didn't bias. We had no bias in any particular population. And now let's look at the genotyping, because that's really what we want to get to. So we did this a couple of ways. I'm going to tell you about the way where we have data, and then I'll tell you at the end the other way that we're looking at this. We first chose SNP marker locations that were relatively equally spaced on the 5 chromosome arms of Drosophila. So we chose this from our significant SNP list, and we tried to pick those SNPs that fell within candidate genes. And these were designed for a TACMAN assay, which is a PCR-based assay. And I'm going to describe that in a minute. So this just shows you the distribution of marker alleles in our most extreme lines. So again, our long sleepers here are at the top and our short sleepers are at the bottom. So if we look at the flies that have the major allele versus the minor allele, you can see that the long sleepers tend to have the green major allele and the short sleepers tend to have the blue minor allele. So that'll be relevant in a second also. But to do the TACMAN assay, you can design DNA probes that will anneal to your SNPs. So in this case, the major allele is A, and we have a probe with a T on it that will anneal to this particular A. And this probe will anneal to the minor allele. Each of these probes has a fluorophore on it. This one has the VIC, the green fluorophore. This one has the F, the FAM fluorophore, and it has a quencher on it also. So that means that when you put these probes into a solution, then you don't really have any fluorescence. And then as PCR proceeds, this probe anneals specifically to your SNP, because you've got FAM in here too. And then hopefully what you see is a lot of green fluorescence in this case. So we do this on a grand scale with the Fluidigm microfluidic chip. This allows us to put DNA from 96 individual flies into one side of the chip, and our TACMAN assay from 96 probes and primers into the other side. These wells here, each one is 9 nanoliters. So what we get out of it is about 10,000 genotypes. And we can pick out the homozygous major, homozygous minor, and heterozygous genotypes from that. So this is what our allele frequencies were prior to selection in the DGRP parental lines and in our mixed population. And so here I'm just showing you, I don't have my numbers here, but on the y-axis it really goes from 0 to 100% in 20% increments. So you can see that the vast majority of the each of our SNPs, which is each vertical line, is the major allele. That's why it's the major allele. And the blue minor allele takes up the rest. And they're very similar between the parental lines and that initial population. But of course, in our mixed population, if we look at the genotypes now, which I'm showing you in the homozygous major in the green, the homozygous minor in blue, and the purple heterozygote in purple, you can see, of course, we have many more heterozygotes, and that reflects all of that mixing. So now we have to deal with heterozygotes. Life's not easy anymore. So let's look at the phenotypic response to selection. Our night sleep selection here shows you the generations from 0 to 12. So we've done 12 generations of selection. The long sleepers here sleep a lot longer. The short sleeper populations sleep a lot less, and our controls stay relatively flat. For day sleep, we see the same trend. The long sleepers sleep longer, the short sleepers sleep less, and the controls are flat. So I just want to quantify this for you because it's quite extreme. I was really pleased with the way this turned out. The long sleepers sleep about 3.5 hours more after 12 generations, and the short sleepers sleep about 7 hours less. So, in all, we've got about 10 hours difference between the long sleeper and short sleeper lines. So that's quite remarkable. For daytime sleep, because we have a positive genetic correlation between night sleep and day sleep, we did expect to see the correlated response to selection. So day sleep does increase and night sleep does decrease, although not so much for this replicate. We saw an extreme decrease in the phenotypic variance, and you sort of expect that in a selection experiment. You're basically inbreeding, so you're taking a small bit of the population and using it as parents each time. So you expect that to decrease, and it does for the long sleeper lines that I'm showing you here, almost according to theoretical prediction, which is by these dotted lines. The short sleepers, however, did not decrease so much. And I think that's a reflection of that variability that we saw earlier among the individual flies. So the short sleepers are quite interesting. We did see very little evidence, however, of inbreeding. All this graph is meant to show you, feeling a little nervous here because I'm running out of time, but all this graph is meant to show you is that our numbers of flies that were viable really didn't decrease over generations of selection, even though we're inbreeding. So let's look at the genetic response to selection. These populations can be subject to genetic drift, which is simply a random change in the genetic composition of our population over time. So if we have a red homozygote, blue homozygote, and purple heterozygote, just from random chance after one generation, you can get a decrease in the blue homozygote. That goes on long enough, the population goes to fixation. And it doesn't matter what fly mates with another, as long as there's no mutation or new migration into that population, then you're not going to change this allele. It's fixed. And the smaller the population size we have, the more you expect, the faster the fixation will take place. And so we looked at our markers here, and we're in good shape. We don't really expect genetic drift to be a factor until 15 generations of selection. And I was just telling you that we've done 12, so we're not quite at the point yet where we'll see that. So this is the results that we have for our genotypes. This is the long sleepers at generation 0, 5, and 10. And again, I'm showing you the percentages here of the major, minor, and heterozygous alleles, or genotypes, rather. And you can see here that the long sleepers tend to get more heterozygous. And I think that's a reflection of the fact that, once again, we saw those major alleles being more prevalent in the long sleepers, as well as possible dominance. So the heterozygotes could be dominant. In our short sleepers, the situation's a little bit more strange. We actually have quite a few more genotypes to go, but we saw quite a few minor alleles in the beginning. They kind of went away by generation 5, and by generation 10, they're coming back. I can only say that we can't exactly explain this yet because we still have about 138,000 genotypes to resolve. So I think that numbers here might be part of the story. And we saw something similar with the controls, where the minor alleles decreased a little bit and then kind of came back in generation 10. So if you do these calculations numerically, we found 6 SNPs that were significantly different between the short sleeper and control and the long sleeper and control. And those genes fall into this particular gene for long sleep. It's in the 5' UTR and the function's unknown. But we found also for short sleep a SNP in the coding region, 3 SNPs in introns. One's in a transcription factor, one is a gene involved in axon guidance, and one's involved in cell adhesion. And then another gene which has an unknown function. There's a SNP upstream of this that's significantly associated with a short sleep. So we found these large changes in sleep phenotypes within a few generations. It's really remarkable, but it seems like the genotyping is a little bit more subtle. And I told you we're going to do this 2 ways. So one way is the genotyping using the PCR-based assay, but the other way is by whole genome sequencing. So we collected pools of flies at every generation, and we're going to look at that. That allows us to look at every SNP in the genome, but the difference is that we can't associate the SNP to the sleep in a single fly. We have to associate it to an average of flies. Dominant alleles could also be present, and we're following up on that analysis. And it could be that haplotypes are actually important here, that epistasis could be acting between the marker alleles. So that's everything. I did want to thank the people who are doing the work. So this is Yasmin Serrano-Negron. She's my technician in the lab, and she's done the lion's share of all that fly work that I just showed you and the genotyping. Amanda did the viability study. So thank you very much.
Anyway, could you explain in the human homologs, or I guess, the human homologs, are they associated with sort of clear-cut phenotypes?
Are the human homologs associated with clear-cut phenotypes in humans? Yeah. So these, are you talking about? I'll go through. This one's long. There it is. No. So no, these are polymorphisms, and these are candidate genes. So several of these are actually in between genes, and these are the closest loci to them, right? And there's one, I think this one is in an intron. So the change in protein function is unknown at this point. It doesn't mean that it's not doing anything, but that's what makes it so difficult in humans. You find this association is statistical, right? So you don't really know what the causal locus is. So that's what we're trying to do in flies. And I should mention that, you know, in humans, we're not looking at all of the gene sequence. We're looking at a chip that genotypes, that has SNP genotypes across the genome. So we're not getting every SNP. So that means necessarily that there's probably linkage disequilibrium between the chip SNP and the actual causal SNP, right? In flies, the situation is different. There's very little linkage disequilibrium in flies. It actually decays over about 10 to 30 base pairs, which is unbelievable. So it means if you find a SNP inside a candidate gene, that should be your causal SNP.
So are we to interpret that somebody looked at humans with these various sleep traits?
Yes.
And then did SNP analysis?
Yes.
And that's what they found?
Yes.
So in other words,
now they're picking.
My daytime sleepiness has a good chance that I have a PD or the SNP association, which may be connected to some gene.
Yeah. Okay, so you're asking if there's a SNP. I'm sorry, I'm not repeating. So you're asking if there's a SNP association in some gene with daytime sleepiness. That's correct. We also found one in this same candidate gene in melanogaster. So this is a cyclic AMP phosphodiesterase. And so that has something to do with the cyclic AMP signaling, which is stimulated, at least in humans, by caffeine. So there could easily be a relationship there.
Anybody have any questions?
Are there other ways in which you define sleeping other than moving? For example, you could be selecting for other physical deficiency rather than sleep. How do you know it's sleep and not just motion that you're measuring?
So one of the things we do is we look at other parameters. Waking activity is a really important one. So if we can show that the fly's movement during the day is not related to sleep, then we feel more confident that we're looking at sleep and not activity. And that's the case. There's no correlation between waking activity and these sleep parameters that I was telling you about, the sleep duration and the sleep bout length. There's another aspect. Oh, everybody's looking for that. Everybody wants this. We've touched on it several times in this discussion, but that correlates with mine. So the only one that I know of is one that Paul Shaw's group has come up with, and that's amylase. And they see it both in flies and in humans. So there's an upregulation of amylase when you're awake, and then it goes down when you're asleep. And the same thing happens with humans. So they show that during waking or sleep deprivation, the levels of amylase in the saliva of humans go up, and then it comes down during sleep. So that's one potential biomarker. But everyone's looking for a biomarker of the type that you described. Can they what?
Are they defining sleep, and are they defining sleep by walking up and down the tube or some other way?
In humans, or in the flies? So for that particular study, oh yes, the same way in the flies using the trikinetics. Yeah. Well, I hope so.
Any other questions? Okay. Yeah.
So I'm truly the layman. So I gather what you're trying to do is you're trying to identify genetic markers in the human genome that then you could develop treatments for disorders.
That's the general idea. So we start with flies, then we have to show that the genes that we find have homologous function in humans, and then we have candidates to go after potentially for sleep disorders. That's correct. takes a while to get there, though.
So what
So
happened?
I just wanted to say one other thing about this activity assay. So I mentioned before that 5 minutes without an activity count was defined as sleep. So what people did to show that was they waited until they watched the flies, waited until they had rested for 1 minute, and then stimulated them. And they respond. So they had their same arousal threshold. The same was true for 2 minutes, 3 minutes, 4 minutes. By 5 minutes, they weren't responding. So they had the increased arousal threshold. You really have to smack on the tube to get them to arouse. So that was one of the ways that we show that the flies are not just sitting there quiescent. They're not just sitting quietly, as it were. They are actually sleeping.
How do you visualize where studies in Drosophila might impact the question of why do we have sleep?
I missed the first part.
Why sleep? How do you have some ideas as to how studies in flies may impact on the question of why do all species sleep?
So with what we have found so far, one very interesting thing is that we found a lot of genes that have to do with protein turnover, which I think is remarkable. So that supports this idea that there are some changes in protein synthesis that are taking place during sleep and wake. Another is different levels of neurotransmitters. Although we're not measuring their levels, with the genome-wide association, we find different SNPs in neurotransmitters like the dopamine and serotonin receptors, for example. And then finally, there's also an interesting class of genes that came up in this screen that have to do with central nervous system development. And that begs the question is, does your brain architecture determine how much you'll sleep? Do you need it based on that? Or is it something that these genes are doing at the adult stage? And we don't have the answer to that question yet, but I think it's really intriguing.
Okay. All right. Well, listen, thank you both very much. That was very exciting. That's marvelous.